Assalamualaikum warahmatullahi wabarakatuh. My name is Syazwin Aqilah binti Azman. So today I would like to share with all of you about ethical issues of genetic technologies. As the new age of biotechnology begins to have its impact on modern societies in various ways, the application of such technologies in certain areas of human activities is being questioned from an ethical standpoint. Although genetic technologies have a great potential to change the medical practice as we know it, it also has the potential to be misused and lead to further health disparities, discrimination, and inequality in human societies around the world. There are many new genetic technologies being introduced in the world on a monthly basis. However, a few have been recognized to have a particular consequence for human societies across the world. These new genetic technologies are believed to have the potential to alter human nature and society at the most fundamental levels. There are three main categories of concern regarding the application of genetic technologies. First is human cloning, second is genetic trait selection, and the last one is genetic modification. Human cloning refers to the creation of either human embryos or human children that are genetically identical to their living or dead parents. Genetic trait selection refers to selection of sperm, eggs, or embryos that possess genes which are associated with certain traits. Genetic modification refers to changes and manipulation of genes in living human cells. For human cloning, the two different applications of cloning technologies are research cloning and reproductive cloning. In research cloning, clonal human embryo is used for experimental purposes. Research cloning is also known as somatic cell nuclear transfer. In somatic cell nuclear transfer process, a nucleus from a somatic cell such as skin or muscle cell is transferred into a female egg from which the genetic material has been removed. At the end of this process, a clonal embryo is produced. For reproductive clonal refers to creation of a clonal human embryo, but instead of being used for experimental purposes, it would be implanted in woman's womb and brought to term as a born child. Embryonic stem cell research in some forms involve research cloning. Therefore, it must be considered in the cloning debate since it does not involve modification or trait selection of genes. Human reproductive cloning remains universally condemned, primarily for the psychological, social, and physiological risks associated with cloning. A cloned embryo intended for implantation into a womb requires thorough molecular testing to fully determine whether an embryo is healthy and whether the cloning process is complete. In addition, as demonstrated by 100 failed attempts to generate a clone macaque in 2007, a viable pregnancy is not guaranteed because the risks associated with reproductive cloning in humans introduce a very high likelihood of loss of life. The process is considered unethical. There are other philosophical issues that also have been raised concerning the nature of reproduction and human identity that reproductive cloning might violate. Concerns about eugenics, the once popular notion that the human species could be improved through the selection of individuals possessing desired traits also have surfaced since cloning could be used to breed better humans, thus violating principles of human dignity, freedom, and equality. The study performed by researchers at the Whitehead Institute for Biomedical Research in Boston found that cloning to create new animals will almost always create an abnormal creature. The researchers looked at 10,000 genes, making it the largest study of its kind. A preliminary study of just 12 genes, as well as circumstantial evidence such as Dolly the clone sheep's arthritis and various clone stillbirth, early aging, and other health problems had already suggested problems with cloning. This larger study found that 1 in every 25 genes was abnormal in placentas from cloned mice. The livers of the cloned mice also showed less serious genetic problems. In conclusion, the data reveal high rates of abnormalities in the cloned animals of multiple mammalian species and lead the panel to conclude that reproductive cloning of humans is not now safe. The sacrifice impact of countless children to death and physical abnormalities as well as mental illness cannot be played out for the sake of science. So that's all for me. Thank you.